Yeah, let's begin. We've already mentioned Gerd Müller, but a good Austrian evolutionary biologist. 2-3 years ago, he kicked off this conference at the Royal Society in London on new trends in evolutionary theory. And it was a conference called by leading evolutionary biologists who were dissatisfied with the standard neo-Darwinian approach to the origin of biological form. And Müller kicked off the conference by enumerating the explanatory deficits of what he called the modern synthesis. And this highlights something about any research program, and that part of what a good research program does is provide explanations for already known facts. And Müller was unsatisfied with the explanatory power of the neo-Darwinian approach and enumerated 5 or 6 key objections or problems, unsolved problems, one of which was the origin of phenotypic complexity and phenotypic traits. The other was the origin of organismal form in the sense of novel body plans. He acknowledged that mutation and selection can explain small-scale modifications and variations, but not large-scale innovation. And then he also highlighted the problem of the discontinuous origin of novel plant and animal body plans in the history of life in the fossil record, and a number of other things. It was a very sort of candid conference in that you had a lot of evolutionary biologists expressing frustration at the dominant view, but at the end of it, as they were there gathered to propose new evolutionary mechanisms that might complement, supplement, or replace the mutation selection mechanism of neo-Darwinism, even one of the key organizers, Susan Mazur, said the conference was characterized by a lack of momentousness, that the problems were well characterized, but no real breakthroughs had been made with respect to the crucial problem of the origin of biological form. As it happens, many years before, I had attended another conference that was not about biological evolution, but more about chemical evolution and the origin of the first life. And there I first encountered the authors Charles Thaxton, Walter Bradley, and Roger Olson, Dean Kenyon, a leader in the early days of the intelligent design movement. These were scientists who were working on the problem of the origin of life, who had become very dissatisfied with chemical evolutionary theory as an explanation for where life had come from out of a prebiotic environment. And the key question that they encountered addressing these explanatory deficits was the problem of the origin of information. That they realized that to build living cells, you needed proteins, but to build the proteins, you needed genetic information. And so origin of life research by the mid-'80s, late 80s, had come to a kind of impasse where leading people in the field recognized, again, explanatory deficits. The explanatory framework that was provided to that point was not sufficient to explain the key facts, one of which was this problem of the origin of information. So in looking at intelligent design as an alternative, one of the first things we want to do is address the question of whether or not intelligent design can provide an adequate explanatory hypothesis. Does it provide an explanatory framework? We'll see a little bit later in the talk that good scientific research programs not only explain already known facts, or they explain already known facts, but then they also may lead us to discover new facts or make predictions about what we ought to find in nature. So these are kind of 2 facets of a research program. I want to start the talk by first sketching the logic of intelligent design as an explanatory hypothesis. When I first encountered Charles Thaxton, I got to know him pretty well over the first year or so after this conference where I met him. And we had long conversations about the thesis of his book, The Mystery of Life's Origin, which was mainly a critique of then standard chemical evolutionary models for the origin of life. But Thaxton and his co-authors had also proposed in an epilogue kind of radical, not completely new, but a radical reaffirmation of an old idea, which is that maybe what we're looking at in biological systems is actual evidence of design, not just the apparent design that the Darwinians talk about. And in particular, they were seized by the informational properties of DNA. If DNA is really an information-bearing molecule, and if that information is in some way functional, doesn't that suggest, they argued, some need for perhaps what they called an intelligent cause or mind? Intuitively, they argued that information is a mind in our experience, it's a property of minds, not of undirected material processes. So they proposed in this sort of, they called it a philosophical epilogue. They sequestered it from the main scientific argumentation of the book, but they did propose this provocative idea. And I had long conversations with Thaxton before going off to grad school. I wasn't entirely convinced of his idea that biological systems were giving evidence of an intelligent cause, but I was really intrigued. And I had a kind of burning question, which was, could this design hypothesis that they were formulating based on the information-bearing properties of DNA be rendered or formulated as a rigorous scientific argument or a rigorous scientific inference? And that led me to want to clarify two questions. First of all, when they talked about information, what kind of information were they talking about in biology? A lot of their argument was followed in the wake of the molecular biological revolution, which started in the 1950s with Watson and Crick and the elucidation of the structure of the DNA molecule. But also, I think a crucial breakthrough in our understanding of what DNA was doing was the 1957 sequence hypothesis of Crick, where he formulated as a hypothesis the idea that the nucleotide bases along the spine of the DNA molecule are functioning like alphabetic characters in a written text or like digital characters in a section of machine code. That is to say that it's not the physical or chemical properties of those chemical subunits themselves that matter to the function of the DNA molecule, but rather their arrangement in accord with an independent symbol convention later characterized as the genetic code. And there's a fascinating story from about 57 to 65 when Crick's sequence hypothesis was essentially validated by work on both sides of the Atlantic in that frenzy of activity we call the molecular biological revolution, where after that period of time they were able to map what's called the gene expression system. Crick was right. DNA is encoding information in a digital form, and that information is functional. So one of the first things I needed to clarify in my thinking about this possible design hypothesis is what is it precisely that needs to be explained. If we're talking about information, it doesn't appear that we're talking about mere Shannon information. The mathematical theory of information that Claude Shannon developed was unable to distinguish between information that was, or a sequence of characters that was merely improbable to a calculable degree, and a similarly length, a sequence of similar length that was also functional. And I quickly, by digging into this a bit more, realized that the biologists, particularly Crick, who was very sensitive to these issues in information sciences, was affirming that the kind of information we're talking about is functional information. It's, in fact, what he wanted to make clear was that the arrangement of the characters is critical to the overall function of the molecules. And so he says information means here the precise determination of sequence, either of bases in the nucleic acid or amino acid residues in the protein. And so this gave rise, and this was a distinction that was in the original Thaxton and Bradley book, between mere complexity or Shannon information, as exemplified by the top string characters, and specified complexity, or as exemplified by the bottom string. So that helped me a bit. Now I knew what was needed to be explained in a biological context. It was specified complexity, or what we've called functional information or specified information, not mere Shannon information. But as I kind of took a deeper dive on this question of could this be formulated, could this design intuition be formulated as a rigorous argument, one of the other people that helped me was another Charles, and that was Charles Darwin. So I got to grad school, I started studying the method of reasoning that historical scientists use to investigate questions in the remote past. And I discovered that Darwin used a method known as inference to the best explanation, or sometimes called the method of multiple competing hypotheses. And I was fortunate in my last year at Cambridge to intersect with a young American professor who came to take a position at Cambridge named Peter Lipton, later rose to become the chair of the department. And Lipton had just written an interesting book on this very method of reasoning called inference to the best explanation. And philosophers who had addressed this question had raised a critical question. If we're comparing competing hypotheses, we want to infer that one which, if true, would best explain the evidence. On what basis do we adjudicate the question of best explanation. What does it mean to be a best explanation? What's the criterion by which we judge? And it turned out that yet another Charles crossed my path, and that was Darwin's mentor, Charles Lyell, or one of his mentors. And in the book, The Principles of Geology, Lyell had a very practical criterion by which he was able to assess competing causal explanations. And that was the idea of causal adequacy, or as he put it, when we're trying to explain an event in the remote past, we want to look for causes that are now in operation. This was also the idea behind his uniformitarian dictum that the present is the key to the past, that our present knowledge of cause and effect should help us adjudicate competing causal hypotheses or explanations. So, for example, if we want to explain an event in the remote past, we might posit various types of causes to explain that event. I use an example with my students of the white powdery stuff that for quite a long time after May 18th, 1980, still graced the rolling Palouse country in Washington State in the United States. If you weren't around on May 18th, 1980 and didn't know about the Mount St. Helens eruption, you would have to use an historical scientific method, almost a forensic method, to figure out what caused that white powder, those patches of white powdery stuff. And so if you were using Darwin's method, or Lyell's, you would say, well, let's posit some possible causal explanations. Maybe it was a storm, maybe it was an earthquake, maybe it was a flood, or maybe it was a volcanic eruption. Not having seen the event itself, but reasoning historically, forensically, which of those four explanations would be best. Well, on Lyell's criterion, it would be the volcanic eruption, not because you saw it happen, but because you had seen previous volcanic eruptions produce that same kind of effect. It was that volcanoes were causes now in operation, or presently acting causes. Darwin called this criterion the vera causa criterion, the idea of we want a true cause, a cause known to be able to produce the effect in question as a good or best possible explanation. So in the best of cases, this method is eliminative. You look at the other possible causes. If they don't have verifiable causal adequacy, we don't have observable evidence of their causal adequacy or theoretical grounds for thinking that a positive cause could produce the effect in question, we can eliminate them from consideration, at least provisionally, and then we elect the cause or causes as either a best or better explanation than those that lack that causal adequacy. Now, I was thinking about all this and thinking still about this question of the origin of information, functional information, when I came across a passage in a small volume that was applying the information sciences to molecular biology. It was written by an early pioneer in this field named Henry Quasler. And on page 16 of this little volume, this quote jumped out at me. He said, "The creation of new information is habitually associated with conscious activity." I thought, that's the Lyellian dictum right there. Habitually associated is uniform and repeated experience, or a cause now in operation. And I thought, well, conscious activity is intelligence, it's mind, it's agency in action, and that is a cause of which we know that can generate information. And so I thought, well, that means that at least one possible cause for the origin of information is mind. And, well, then it took me a while to work out the rest of this, but I wrote a big book called Signature in the Self. And what I did was I applied self-consciously this method of multiple competing hypotheses to different explanations for the origin of the information necessary to build the first cell. And as you look at the history of ideas about the evolutionary abiogenesis, you have explanations that rely on chance or stochastic processes. You have explanations that rely on necessity or law-like processes. You have explanations that combine the two. And then there was this concept that I was considering, which was the idea of an intelligent cause or mind or conscious activity as a possible scientific explanation for the origin of specified complexity or information. Now, some of you know about the work I've done on this and other design proponents and work that they've done, but essentially what I want you to see tonight is not, we're not going to do an evaluation of all these different classes of explanation. But what I did in the book was very methodically evaluate the key chemical evolutionary ideas as they exemplified one of these 3 or 4 different approaches to explanation. I found 3 of them inadequate for, in some cases, very profound theoretical as well as empirical reasons, and yet, at the end, concluded there is a cause of which we know that is capable of generating information, especially in a digital form, and that is, again, mind or intelligence. So using the very method that Darwin pioneered, I concluded that there was a positive basis for an inference to intelligent design as a matter of historical science, using a standard historical scientific method. And so when we think about the theory of intelligent design, we often define the theory using this best explanation language. We're building something of our historical scientific method into our understanding of what the theory is affirming. And here's a definition we use: that key features of living systems can be best explained by the activity of a designing intelligence rather than an undirected process such as natural selection acting on random variation or other undirected natural processes. And therefore we can say things like living systems look designed because they were designed. And that is obviously contrasting our point of view, not with, well, many other things, many other aspects of evolutionary biology we're not taking on, but we are directly contrasting our perspective with this claim of the staunch Darwinians and neo-Darwinians who claim that biology is the study of complicated things that only give the appearance of having been designed for a purpose. And that's the reason that intelligent design was named intelligent design. We were contrasting the design hypothesis with the apparent design hypothesis of those of a Darwinian persuasion. Now, many other ID arguments can be understood in light of this same kind of best explanation logic, okay? And it'd be fun to go through them all. We don't have time to do that, but we can give a list. The anthropic fine-tuning argument is, I think, best formulated as an inference to the best explanation, where we're comparing the causal adequacy of other explanations for the fine-tuning, like the weak anthropic principle, which actually fails for the causal adequacy test, or the multiverse, and we can get into that. Michael Behe's argument from irreducible complexity is identifying another feature of living systems that requires explanation. Sometimes we might think of it as the functional integration of parts, or what he calls irreducible complexity, whether it's of molecular machinery or circuitry in cells. And he argues for intelligent design as the best explanation for that feature, based again on our uniform and repeated experience of what it takes to generate that feature, or based on our cause and effect knowledge of the world. Whenever we see systems that are irreducibly complex and we know the causal story behind their origin, invariably we trace them back to a mind, typically of an engineer, when we're talking about this type of complexity. I wrote another book extending the information argument, which was based on the Cambrian explosion and what I characterize as an information explosion, and showed that the ID hypothesis also can be formulated as an alternative hypothesis to various evolutionary explanations for the origin of new forms of life, even if we presuppose pre-existing forms of life. So the ID hypothesis can be an explanation, can be an alternative to chemical evolutionary theory or various forms of biological evolutionary theory. And some of us will be gathering last day of this conference to talk about a new project that we're pulling together called The Unexpected Genome, which is just sketching the broad explanatory power of the ID hypothesis vis-a-vis standard evolutionary models with respect to many different classes of evidence that are coming to the fore, whether it's orphan genes or overlapping genes, or the unexpected rarity of genes and proteins in sequence space, or the unexpectedly long waiting times associated with key evolutionary transitions. Where the biggest problem we have with this book is the number of chapters. We're up around 40 different anomalous features of biological systems, especially related to their informational properties or features that are unexpected on a neo-Darwinian view and other undirected models of evolution, but would be expected if we take agency or design, or what agents can do seriously as a possible explanation. And these arguments using the best explanation logic can be reinforced by, for example, the formalism that William Dembski developed for, in his theory of design detection. They may also be reinforced by considerations from epistemology, like the use of Bayesian logic or Bayesian probabilities. So the best explanation logic is consistent with a lot of other ways or epistemological tools for evaluating competing hypotheses. And so that's kind of a sketch of intelligent design as a research program. Now, in the interest of time, I'm going to skip over a little bit of this, but you can see that with these other hypotheses, like the fine-tuning, you can give kind of a cognitive map of the terrain, the competing hypotheses. I like this one: the strong anthropic principle, the weak anthropic principle, fundamental laws of physics, chance, in this case meaning the multiverse, and design. And I'm writing on this now and using the same kind of approach to this, evaluating the causal adequacy of these competing hypotheses. And this is what I did. This is what I did in also in Darwin's Doubt, looking at the discontinuous increases of biological information after the origin of life, in particular in events such as the Cambrian explosion, which I characterize as an information explosion. There we had a lot more different hypotheses. In fact, there are so many new post-Darwinian evolutionary theories now, it was necessary to really look at a lot of different models. But what was interesting about that, and what I found, was that invariably, these new models, even if they captured some aspect of biological reality that the neo-Darwinian theory didn't, they were still subject to the information problem. They either didn't address the information problem, or they only succeeded in solving it by presupposing prior unexplained sources of functional biological information, either at the genetic or at the epi or ontogenetic levels. So that was a kind of a fascinating discovery in doing the work on this. And it was underscored by an interesting critique I got of the book. And this was from Charles Marshall in Science. He did me the honor of a critical review in Science, which I appreciated, but I especially appreciated it because of the critique itself. He said that he correctly represented my view as making an information-based argument for design. He said that Meyer's case depends on the claim that the origin of new animal body plans requires vast amounts of novel genetic information. That is what I argued. And then he said, but in fact, our present understanding of morphogenesis indicates that new phyla, the new animals, were not made by new genes, but largely emerged through the rewiring of gene regulatory networks of already or acting on already existing genes. This was a fascinating critique because he obviously is begging the question as to multiple sources of new genetic information, or of pre-existing genetic information. Gene regulatory networks are chock full of different genes and their gene products. They control animal development. They act on other genes for building the body parts of animals. That's another source of pre-existing genetic information. And he'd already explained in his technical work that these gene regulatory networks are highly resistant to perturbation. And so to get one to change and maintain function, you would need multiple coordinated changes in code. So in this short sentence critiquing the information-based argument I was making, he didn't solve the information problem, but rather he presupposed prior unexplained sources of genetic information. And I've seen that happen repeatedly in the debate about the origin of information, which leads me to suspect that we're onto something very fundamental in the ID movement. And so one of the things about information, or sorry, about research programs is that and the best explanation strategy for advancing them is that the best explanation argument is always provisional because it's scientific. We don't produce deductive proofs in science with 100% certainty. But that doesn't mean we need to wait forever to get that either. If we have a good rationale for affirming a particular explanatory hypothesis, and if in particular attempts to refute it only sort of reinforce it, as Marshall's critique I think does of the information-based argument, it may be time to start using that explanatory hypothesis to generate new testable models or predictions, or in other words, using your explanatory hypothesis to guide further research. And that's the exciting recent development in the ID research program that's starting to unfold. Is we've got a lot of really exciting new research programs that aren't so much trying to make the case for intelligent design, but rather to use intelligent design as a heuristic or a guide to discovery. So a good scientific research program in general, this is just kind of straightforward philosophy of science now. Emir Lakatos, Polanyi, lots of philosophers of science have seen this, that good scientific research programs provide better explanations of known facts. They do that by providing positive evidence for the causal power of a postulated entity and by marshalling evidence against the causal power of some alternative proposed hypothesis or competing hypotheses. But in addition, and this is what the second part of it, of a good research program, that that hypothesis can then be used, or its conceptual framework can be used to generate predictions about as yet unobserved facts, what we ought to expect to see, for example, in living systems, since we're talking biologically, or in physics, in any field. And that's the second facet of a good research program. Now, one way that this can play out in the case of intelligent design, one kind of logic that helps us generate testable predictions or expectations about the living world is a kind of logic that plays off of our awareness of the functional logic of living systems. My colleague Paul Nelson calls this design triangulation. And he thinks of it as a metaphor to geometry at first. If we know, the angle A and we know the angle B, we can infer or deduce the angle C. In biology, we can do something similar to that if we're aware of an overarching functional logic. And we know something about what some of the parts do of the system, but we don't know what all of the parts do. We can often infer that there must be something else out there that we haven't discovered yet that will complete the picture, that will show how the function overall is performed. He has a nice example from some recent literature where we conjoin two facts. We know that free copper is highly toxic to cells. We know that copper is nevertheless absolutely required by aerobic organisms. And so that led researchers to infer there's got to be something else out there that allows the copper to be used without it being harmful to the organism. In other words, there must be a cellular system that must exist that binds, transports, and releases copper where the metal is needed. Now, I had to cut a bunch of slides. This is a really cool example, and this mode of reasoning was used to make a discovery. Now, so a discovery about life that wouldn't have been discovered otherwise. Now, it may be that someone could make that prediction not being aware that they're actually employing design reasoning, but that's what they're doing. And this is Paul Nelson's point. He says, when one discovers a complex system performing specialized functions, one should infer that a rational logic and a set of well-matched parts are providing the functions, even if one has not yet observed those parts or inferred the logic. Now, why is that the case? Well, again, it's because of our uniform experience. Our universal experience shows that specialized functions require rational foresight, design, and then an agency to actualize the design plan. And so when we observe such functions in play, we can be strongly confident that the active features, the parts, and the functional logic are present, even if we haven't discovered them yet. And so there's a kind of, in this use of functional logic to generate predictions about what might be out there, there's an implicit design reasoning underlying it. And even scientists who aren't explicitly ID friendly, maybe even if they're ID hostile, have used this mode of reasoning to make discoveries. And I think we can use it being explicitly ID friendly. Let me tell you just one more fascinating example of this. Watson, well, it's not so much Watson, it's Crick. And this is Crick's genius. He was, by the way, he's a code breaker in World War II, which I think is really kind of fascinating given the discoveries that he made. But you may remember his famous adapter hypothesis about the transfer RNA, about tRNA. And what Crick realized was that the chemical structure of the bases in the DNA were so different than the chemical structure of the amino acids that he didn't see how DNA could function as a direct template for the sequencing of the amino acids. So instead, he anticipated that there must be something that is adapting or translating that information. And so here was a quote from him in 1955. He says, "Now what I find profoundly disturbing is that I cannot conceive of any structure for either nucleic acid acting as a direct template for amino acids, or at least as a specific template. I don't think anybody looking at RNA or DNA would think of them as templates for amino acids. The chemical structures are just too different. And so he ends up doing this, he does this design triangulation. Here's how he reasons. We've got DNA carrying information, but in a chemically non-specific form. We have eventually proteins being built out of amino acids, yet the amino acids have very specific geometries. Therefore, he concludes, wrong way. Therefore, he concludes there must be a mediating or adapter molecule that exists, or rather molecules, that enables specifying that allows us to or allows the cell to specify the information to pass from DNA to amino acids. And he predicts the existence of this molecule. And here's how he boldly triangulates. He says each amino acid would combine chemically at a specific enzyme with a small molecule having a specific hydrogen bonding surface, and would combine specifically with a nucleic acid template. In its simplest form, there would be 20 different kinds of these adapter molecules, one for each amino acid, and 20 different enzymes to help join the amino acid to their adapters. And he went on to describe how Brenner called this the adapter hypothesis. And so here's kind of how it worked. You got the nucleic acid, you've got the amino acids, and we're going to just dial in on one, say valine, and okay, too chemically different to directly template. So we need an adapter. It's got to bind to the amino acid, or to the nucleic acid on one side. It's got to bind to the amino acid on the other, and it's going to need an enzyme to help. And for each one of the 20 different amino acids corresponding to the triplets, you're going to need a specific tRNA and a specific amino acyl tRNA synthetase. And he basically, in the eighth day of creation, Horace Freeland Judson said this was a spectacular piece of reasoning from pure functional necessity to the existence of an entity for which there was not yet a single thread of empirical evidence. And of course, the story is fascinating because he turned out to be right. What is also fascinating is that Watson completely rejected this hypothesis. And he said, and he did so on evolutionary grounds. He said, I do not like the idea at all. More to the point, the adapter mechanism seemed to me too complicated to ever have evolved at the origin of life. It required multiple coordinated interactions of well, at least 20 different enzymes, 20 different tRNAs, the nucleic acid, and then the rest of the gene expression apparatus. And actually, Watson is right about this. To this day, there is no decent explanation, not even anything remotely plausible for explaining the origin of the translation system. It's a huge unsolved problem in origin of life research. This idea was much too complicated to be explained by chemical evolutionary theory. But Crick stuck to his guns on pure functional grounds, on the grounds of the design logic of the system itself, the information processing system. So even though Crick was explicitly a materialist, I want to argue that he was implicitly reasoning like a design theorist. Watson's biological intuition, however, was constrained by evolutionary assumptions, so he rejected the idea out of hand. He had to accept it once the molecules were found, but his initial reaction to this revealed that he was constrained by evolutionary assumptions. Well, let's not be that. Crick was better than his materialistic principles. I would say he was reasoning as an ID theorist, even though he didn't explicitly acknowledge that position. Now, the point is, well, of course, they found the transfer RNA and the proteins. Point is, that's just one logic, one way of thinking about design that can lead to testable predictions. And my first 2 points on this slide kind of unpack this. We use our knowledge of the functional or design logic of biological systems to predict the existence of yet unknown parts or features of organisms. That's design triangulation. But then we can also use our knowledge of what designing minds do and what gradual evolutionary processes would not or are unlikely to do to generate not just predictions, but even discriminating predictions. We can generate predictions that we would expect on a design-based mode of reasoning as opposed to an evolutionary theoretic approach to origins. And so I think there are cases where, for example, we might predict a certain system that would have multiple coordinated interactions of parts that couldn't have been built up gradually. We might need such a system because of the functional logic of the organism that we're looking at, but it's not something that would be plausible on an evolutionary grounds. If we find that, then I would argue we're confirming an ID prediction, an ID-based prediction. But there's other ways in which ID can guide research. We can use our knowledge of the functional or design logic of biological systems to engineer better technology. There's a whole group of biomimetics ID proponents in the UK who are specifically copying what's in nature to build a better mousetrap, or airplane wings, or Jim Tour, who's doing some fascinating work at Rice, who's looking at the design logic of molecular machines and then using his knowledge of that to redesign machines to attack cancer. Really interesting work. Biomimetics. Another way in which ID can guide research is to use engineering principles to explicate the logic of biological systems. We've got Brian Miller is working with a group of biologists and engineers together, and the engineers are bringing a lot of insight into the design logic of systems, whether it's feedback loops or design patterns from computer science that help us to really understand what's going on in biology. And a lot of what's called the field of systems biology is not only, it's increasingly not just implicitly design friendly, it's increasingly explicitly design friendly. Another kind of logic is to use ID to explain the origin of newly discovered features of living systems for which intelligence is the only known cause. This would be extending the explanatory power of the ID program. For example, in Darwin's Doubt, I looked at these developmental gene regulatory networks, which Charles Marshall also referred to. And when I first saw these, they're crucially involved in animal development. When I first saw a depiction of one of these from Eric Davidson, the key researcher on these guys at Caltech, first thing it looked to me like it was an integrated circuit. And in fact, it's an integrated circuit which is controlling the flow of information more than electricity. But one of the things we know about integrated circuits is they're subject to the constraints problem in electrical engineering. The more tightly integrated a system, more difficult it is to perturb it without defect to the whole. These guys pose a big problem to body-plan morphogenesis because you really can't perturb them without breaking them down and causing the organism not to develop and to shutting down the evolutionary process. I would argue that a system like this, because of its functional features, is actually providing additional evidence for intelligent design. It's not just a challenge to neo-Darwinism's account of the origin of body plans. Features that have the character of integrated circuits are systems that are known to have been produced by one and only one type of cause. And therefore, I think when we encounter higher level design like this, when those features map to what philosophers call the explananda, things that need to be explained that we know are only produced by intelligent agents, we may be encountering additional evidence for design and extending the explanatory power of the ID research program. Two more final things. Another thing ID can do as a guide to research, sometimes we can conduct or reinterpret simulation experiments to show the indispensable role of intelligence in simulating evolutionary processes. I'm thinking here of the evolutionary informatics lab of Robert Marks and Bill Dembski. They're actually looking at these evolutionary simulations like EV and Evita, and they're quantifying the input of active information in bits per iteration that's coming from the programmer. So these simulations are presented as providing evidence for the creative power of the mutation selection mechanism, but in fact what they're inadvertently showing is the power of the need for, the indispensable role of intelligence in getting any kind of success in the simulation. And then another area is that when we are using ID to anticipate function, we're actually, this has been very applicable in the junk DNA debate. The non-coding regions of the DNA were immediately affirmed in the 80s as the evidence of that random trial and error process of mutation and selection that the neo-Darwinists had expected as the source of biological information. some of the, Richard Sternberg in particular, but also Dean Kenyon and a few other ID proponents in the late 90s and early 2000s said, well, you know, we would expect something different. We acknowledge that mutations are a real process, but we wouldn't expect that the signal would be dwarfed by the noise. And so we're going to predict that those non-coding regions are going to have important functions. And so there's, I think, a whole class of research, whether it's on junk DNA generally or pseudogenes, that has generated where ID has an expectation that differs from neo-Darwinism, and things like the ENCODE project have confirmed those differing ID expectations. So finally, just a quick list here of some key ID, we call it 3.0 research projects. 1.0 was the critique of Darwinism in books like Darwin on Trial. 2.0 was the Dembski, Behe, and then the many, many different books and articles that have come out advancing ID as an explanatory hypothesis. But 3.0 is now using ID to generate new research. Tomorrow you'll hear about the fascinating work of Siegfried and several others in the room here on overlapping genes, and why overlapping genes are something you might expect to find if the genome were designed, but you certainly wouldn't expect to find if it were the result of undirected processes. And I find that fascinating because the nested coding of information is also a recognized computer design pattern, where a design pattern is an established mode of storing and expressing information in a digital form. And I think what we know about how we human designers design and store information in our high-tech digital computing can also lead to expectations about what we might find in living systems. So we also have a big project on orphan genes and their unexpected pervasiveness across the taxonomic landscape. Orphan genes are genes that don't have discernible sequence similarity to other genes, and yet on a Darwinian or evolutionary model, we ought to expect that sequence similarity in all genes, and yet we're finding wide classes of genes without that kind of similarity, and creating a picture of genetic information almost popping discontinuously into existence across the taxonomic landscape. A search for a new function in a research project on the vitamin C pseudogene that's going on in Israel with a colleague of Tony's, Jeff Barnard. Looking at nano machines, we've got 3 different projects going on right now on nano machines. One on the ATP synthase, one on the bacterioflagellar motor and the co-option hypothesis, looking at which of those 2 provides a better explanation. And then thirdly, the work that Jim Tour is doing on a kind of biomimetics, where he's learning a lot about what it takes to build molecular machines, using that knowledge to critique chemical evolutionary theory, but then also using that knowledge to redesign machines for medical application, in particular a mechanical way of lysing cell membranes in nasty cancer cells. And then we've got a waiting times project we'll hear more about from Gunter and Ola, the informational analysis of evolutionary algorithms I talked about already. And I've been involved in a project that's analogous to that in cosmology, looking at doing an informational analysis of what's called quantum cosmology, showing that models that are attempting to mathematically simulate, if you will, the origin of the universe actually require unexplained sources of information in order to solve the mathematical equations that give you the outcome of a universe like ours. So even in cosmology, there seems to be an informational requirement that pre-exists the origin of the universe, which is kind of fascinating stuff, and we can get into that. So anyway, just this is a real quick kind of list of categories of research that we've got ongoing that's taking ID in a heuristically beneficial direction. So I'll stop there. Thank you.